last but not least, we have Anna Cuomo, who was a graduate student with Ollie Stiegel and John Marioni, but recently transitioned into a postdoctoral fellowship at the Garvan Institute. So she'll be telling us today about how uncovering context-specific and dynamic genetic regulation of gene expression at single-cell resolution. So welcome, Anna.
Okay. Yeah, thanks very much. I'm very excited to be here and to tell you about a tool we recently developed to map context-specific eQTLs in single-cell data. So very, very relevant to the last talk we just heard. And let me start by sharing my own version of this diagram, which we've seen many, many times in this meeting already, including the last talk. And saying that a major challenge in biology is to unravel the genotype-to-phenotype map and understand how DNA variants can shape observable traits and cause complex diseases. And while this is exactly what genome-wide association studies, or GWAS, do, the interpretability of identified associated variants is often far from trivial, especially when we are looking at non-coding variants. And so one way of understanding the mechanisms that link these two parties is to look at intermediate molecular traits, such as gene expression. And as it's been said, the interpretation, sort of the assumption here is that these variants are likely to play more of a regulatory role, like in this cartoon here, where having a G at this specific genomic locus may increase the affinity of a transcription factor to bind and thus boost expression. And methods that aim to find associations between genotype and gene expression are called eQTL, or expression quantitative trait loci, where we look at the expression of the gene of interest on the Y, and this is typically been assayed with bulk, as I'll come back to, and then we stratify our samples by the genotype on the X. And one other aspect I'd like to highlight is that eQTLs are often detected only in some specific circumstances, conditions, and tissues. In one word, they are context-specific. So I'm going to go back to my cartoon here and only show the trends now. There have been many, many examples that have demonstrated how the same SNP or DNA variant can be an eQTL for a gene in one cell type but not another, in activated cells but not naive. And of course, this is especially important if our goal is to identify mechanisms leading to disease. We really want to be looking at the right cell type in the right condition. And the last piece of the puzzle is that in recent years, single-cell technologies, and particularly single-cell RNA-seq, has really revolutionized our ability to characterize the cellular heterogeneity in a number of tissues and systems. And further advances in technology and reduction in cost now allow us to deploy single-cell RNA-seq across hundreds, and as we'll see, even thousands of individuals, which really allows us to start looking at genetic effects on single-cell expression. And in the last 4 to 5 years, there have been quite a lot of, there have been a few single-cell eQTL studies emerging, some of which I've contributed to in my own PhD. And just to say that some of these efforts have been coordinated as part of the Single-Cell eQTL Gen Consortium, whose manifest is here. However, existing strategies to perform genetic analysis using single-cell RNA-seq data largely remain based on principle first established for bulk RNA-seq. So in particular, what one typically does is you have your single-cell experiment and you'll use the sort of full transcriptome to define transcriptionally distinct cell groups and then assign them to cell types and then map eQTL in each of these cell types separately using methods that were originally devised for bulk. And we think this is somewhat limiting and it might miss so sort of more subtle effects either in subpopulations or effects that are more continuous like the dynamic eQTL we just heard about. And so this motivated us to develop a new method that allows us to map context-specific eQTL across a number of contexts and that does not require discretization of single cells into groups. And in the next couple of slides, I'll introduce the method, and I'd like to start by convincing you that what I've previously called a context-specific eQTL is really a special case, what we may call a genotype by context or G by C interaction. And so going back to this eQTL cartoon, I'd like to draw your attention to what we use to quantify the strength of an eQTL, and this is the slope of this line, and I'm calling this beta here. And then within a single-cell experiment, we may have different types of cells, and it is conceivable that a given gene may, the expression of a given gene may change across different types, and in this example, for example, in this cartoon, for example, this gene is more highly expressed in pluripotent cells than differentiated cells on average. However, as long as this only results in sort of a shift upwards of the expression and the slope doesn't change, this does not qualify as an interaction. Instead, we talk about a G/C interaction when the state that one or multiple cells find themselves in actually modulates the strength of the eQTL effect. So in this case here, this SNP actually has a stronger effect on expression in pluripotent cells versus differentiated. And hopefully you can see how the previous examples where one eQTL was only detected in one condition is a special case of this, where the effect is not at all detectable in one condition. However, we argue that there may be more subtle changes that look more like this. And then the next step is that a cell state or identity may actually be defined by multiple contexts and factors that can be measured at single cell level. So in this case, for example, one cell will find itself somewhere along a differentiation state, but also will be in one of the phases of the cell cycle. And those can be both measured in single cells at the same time. And so in this cartoon already, we have 4 different groups of cells and potentially 4 different effect sizes. And hopefully you see where I'm going with this. There'll be many more factors and contexts that can be characterized, and also a lot of these will be better defined as continuums rather than binary, like I've simplified it in this cartoon. And so if you take this to the extreme, really, eventually every single cell will have its own effect size, and that's really the result of the microcellular environment that each cell finds itself in. And so the intuition behind our method is that this effect size is actually a vector rather than a single number that varies as a function of all of these contexts that we can measure. And so that's what we do in our method. So we model the single cell expression of a given gene as a function of a SNP effect. So we do allow the SNP to have a persistent effect or shared across all cells. But in addition to that, we also have an additional SNP effect that is the G/C component that is really driven by all of these contexts. And so we model these using a context-covariance matrix, for which I give an intuition in the next slide. Additionally, we allow cell context to have an effect directly on expression, and that's what was in the top right corner, if you remember the previous slide. Very importantly, in this setting, we have multiple cells coming from the same donor, so it's very important to account for this sort of inherent repeated structure that we have across our samples. And finally, we always allow for measuring noise. And so what this method allows us to do is to test for G/C effects on single cell expression that are beyond G only, so genetic only effects, or context only effects. And as I promised, this is just to give you an intuition of what the cell context covariance matrix may look like. It's a cell-by-cell symmetric matrix, and in the simplified case on the left, where we have only 2 cell groups, this matrix looks something like this, where it's essentially block diagonal, and there's no similarity between the 2 groups. And instead, on the right, we have a perhaps more realistic setting where we have more of a gradient. And I should say this method is now a preprint, so if you want to look at the results, you can check out the paper. And here I'm only going to give one vignette of one result to show what can be done with this. And so what we've done here is we applied the method on data that we actually published last year. And this is a iPSC differentiation experiment where we have from 200 individuals iPSCs differentiating towards neuronal cell types. And here I'm focusing specifically on the dopaminergic neurons. And there were 3 conditions annotated in the original study. We have day 30, sort of younger neurons, day 52, more mature neurons, and then also a stimulated condition. And if we focus on one specific eQTL, we know that this eQTL in the original study was found specifically in this one condition. And now one nice feature of our method is that we can actually estimate this cell level effect size. And so we can color that same TSNA map by the effect size in every single cell. And so what you can nicely see, first of all, is that we recapitulate where the effects are most strongly observed, which is in that day 52 cell population. But additionally, we see a little bit more granularity than that. And so if we now stratify, consider only the cells where these effects were strongest, and remap eQTL in these 2 separate strata, we actually improve both our eQTL signal, and actually this was an eQTL colocalizing with a GWAS hit as well, and the colocalization signal improves as well, despite us losing some power because we're now mapping eQTL with only 1/3 of the cells, and we're losing some donors as well. So it really shows how you can really identify the specific cells where effects are active. Okay, and in the last few minutes, I'd like to present some very preliminary results I have on a dataset I am extremely excited to be working on. So as Jin mentioned, I recently moved to Sydney, Australia for my postdoc, and this is a dataset that my supervisor, Joseph Powell, has generated together with Alex Hewitt at the University of Tasmania. And I'm talking about the 1K cohort, which, as the name suggests, comprises of a bit more than 1,000 cells from each of nearly 1,000 individuals. So this is by far the largest single-cell data set out there, at least in terms of number of individuals. And these are, should say, it's PBMC's healthy individuals and of European ancestry at the moment. And in the first phase of this project, which was published actually just a few weeks ago in Science, so check it out, it's beautiful work that was led by Seyhan and Jose. And what they did is what you do always the first time around, so you use single-cell data to define your cell types, and here they identified these 14 sub-cell types from blood that you can see here, and then map eQTL in each of them separately. And they find more than 20,000 cis-eQTL across the cell types, and confirm that a lot of these are cell type specific as well. So if you want to look at more results, please check the paper, but for now I'm going to focus on the B cells only. And if you subset on only the B cells, you see that we had annotated them as naive and memory, but really we have more of a continuum, as you can see, where cells go from most naive on the right to most memory on the left. And indeed, you can consider a pseudotime. So indeed, in the original paper, to define dynamic eQTLs along this B cell lineage, they had defined a pseudotime. And then they had to discretize along this pseudotime into 6 quantiles, and then map eQTL in each of them. And so this is just an example where you see the effect size of an eQTL in each of these quantiles on the y-axis, and then it's going quantile, so actually it's going right to left in the plot here. And so you see this is an effect, an example where the effect is strongest in naive cells. And again, this is very preliminary results. But when we actually use our method on this data, we can nicely recapitulate. Hopefully you can see how the strongest effects, so the redmost are on the right and then progressively decreases. And obviously this didn't require any pseudotime inference, didn't require any discretization, and could really be run directly from the single cell data. And this is not an isolated cases, but we actually recapitulate every single one of the dynamic eQTLs that were present in the paper, and we actually find a few more. And I'm only showing 4 examples here that are known, there are genes that are known to be involved in B cell activity. And so to conclude, I introduced CellReg Map, which is a method that allows to jointly map multi-context eQTL without requiring discretization of cells into groups. I haven't shown this here, but please check the preprint. We use extended simulations to show that our method outperforms available methods in terms of both power and calibration. This method can help identifying the specific cells where regulation is active by estimating cell-level effect sizes, which can also help characterizing colocalization events. Sorry, just one last slide on future work. So we actually have now collected whole genome sequencing from the data as well, in collaboration with Daniel MacArthur in Sydney. And so this is really the largest data set out there with matched single-cell RNA-seq and whole genome sequencing. So I'm super excited to dive into effects of rare variants as well as common, given the sample size. And then finally, there's a lot of avenues. I see this as one of the first methods for really a toolbox for single-cell population studies, and there's a lot of work to do. For example, I'm looking into implementing non-Gaussian likelihoods, which are known to be better fit for count data. Also needs to be optimized in terms of just the efficiency and the memory usage, because these data sets are getting very, very large. I'm also very interested in looking at effects of environmental and physiological covariates, which we do have access to for this data set, so effectively look for gene-environment interactions and signatures of those at single cell level. Sorry, we're also getting one more cohort that will be of patient data, so that will be also very interesting to look at differences between cases and controls. And finally, the new cohorts that we're getting are also finally not just European, so we'll be able to start looking at ancestry differences as well. So please check out our preprint. The method is implemented as a Python package, so you can also do, you can install it very easily. We have a webpage with a lot of tutorials and instructions, so if you're interested to try it out on your data, please come and find me or check our webpage. I'd like to thank a lot of people. So this was mostly work done in with my previous supervisors, mainly Oliver Stegler and at the EVI in Cambridge, and Danilo and Toby from the lab. I should say this method builds on a previous method from the lab that's called Structured MM. That was work led by Rachel and Paolo. I'd like to thank Nicole Saranza, who very kindly hosted me in the not-so-smooth transition from the UK to Australia in COVID times. And finally, my current supervisors, Joseph Powell and Danny MacArthur. And you for your attention. Thank you.
Wonderful. Questions for Anna? So we have a question here. Yep.
Yeah, thank you very much for the fantastic talk. I was wondering if you have thought about using like some molecular profiles such as ATAC-seq to infer some of the cellular context, like if maybe like complementing some of the samples with a small like multi-omic data set may help to infer the G by E covariate matrix better.
Yeah, that's an excellent question. So I'm going to answer in two parts. So one, obviously this is it's designed to work when you only have a single-cell RNA-seq and genotype. So what we do here is we infer this context directly from RNA-seq, which I agree could be potentially a bit circular. So it would be very nice to have an additional omics to complement it. And we are actually, so for a subset of these samples, we are going to get single-cell ATAC-seq as well. So I'm very excited to dive into that as well. Thank you.
Yeah. Well, we have a question over there.
Thank you. Thank you for this great presentation, a lot of great work.
Thank you.
I'm just curious about your large population single-cell RNA-seq.
Yeah.
Do you align, in order to prepare the data, to pre-process the data, do you align it in a genotype-aware manner, and does it help if you do? Or?
Yeah, it's a good question. So first of all, I should say, in these types of experiments, you typically multiplex samples. So this is in every experiment, you have cells from maybe 12, I think, individuals, maybe in this case. And so you do need to use some tool like the Maxley or Vera to demultiplex and find back. So it's all processed in a very, very similar way. We do have batches, and we correct for that with more standard methods like Harmony. So that's not necessarily genotype aware.
So you're aligned to the reference genome?
Oh, sorry, the SNPs. You're talking about the genotype or the single-cell RNA-seq? Oh, so these are SNP chips, so this is imputed variants separately. Yeah, yes. So it's not imputed from the RNA-seq. Yeah, sorry. Yeah.
Thank you for a really interesting talk. I just had a question about this interesting result of eQTL effect size changing along a trajectory. Mm-hmm. And my question was, do you have an approach to assign statistical significance to sort of monotonicity of such a change?
So the method does come. So what we do at the moment, the method is essentially just testing whether there is any G by C, so that could be with any of the contexts. But we use a score test that gets us a p-value as to where there is, and then I think afterwards to determine specifically which context was driving the signal. I think what we'll have to do, and we haven't implemented it yet, will be to compare a method, sort of a model with and without that one context, and compare.
Good, thank you.
Yeah.
Great. We do have time for one more question. Don't be shy. Just reiterating John's sentiments that we hope that all students will get a chance to ask at least one question before the end of this conference.
Yeah, great talk. So I was just wondering whether you found evolutionary, whether there's difference in terms of their conservation between these context-specific QTL versus those constant QTLs.
Yeah, that's an excellent question, but I have not unfortunately looked at that. Yeah, it's a good suggestion. Thank you.
Perfect. Well, great. Well, please join me again in thanking all the speakers this session. Great. Thank you all. And I would like to ask the speakers to come up for a group photo, but everyone else, please enjoy lunch.